Welcome, Eastview Scholars. Let's talk about macromolecules on this edition of Sharp Shorts. Of course, the first thing to say is the macromolecules that we're going to talk about: proteins, lipids, carbohydrates, nucleic acids. Those are the main large functional molecules that control structure, energy, genetic code, steroid use, or steroid production, hormones, all the things that living organisms need to keep working. 40 to 50 common monomers, carbon skeleton and functional groups with them. Okay, and when you put them together, it's a condensation or a dehydration synthesis. To form a polymer, you put monomers together. When you break them down, or hydrolysis, split by water, then you break them into their separate monomers. So, you have catabolic and anabolic reactions going on all the time. You have catabolic is breaking down. So the foodstuffs you bring in and eat, you're breaking those into the monomers that they are. For example, the sugars, the carbohydrates you're breaking them into the monomers to get glucose for your cell to use to provide energy. And then the anabolic portion is that energy that is derived is then used to put monomers back together, amino acids to make proteins, etc. Okay? So, and here's just a simple example of that. Here's a dehydration synthesis. You're making a polymer, where here, as you're breaking apart, it's the hydrolysis portion. Okay, so again, 40 to 50 monomers. Water is a key molecule. Here are the macromolecules we're looking at. Fats or lipids, carbohydrates, proteins, nucleic acids. Let's start with the carbohydrates. These are your sugars. Now, all the macromolecules have multiple functions, but they all generally have one main function. And for carbohydrates, it's energy. These are the sugars. They all end in an ose. You can see here ribose, glucose, galactose, fructose, sucrose. Sometimes they're ketones or aldehydes and stuff. That's functional group stuff that we talked about before. But the main function here is that you break them down and use the glucose and stuff to provide you energy and to drive the synthesis of ATP and cell respiration, which we'll talk about later. Carbohydrates also have a structural function on the outside of human cells. They are part of what's called a glycocalyx, and that identifies your cells as yours for your immune system. Okay? There's both linear and ring forms. Mostly you see ring structures. Okay? And again, here is where we're pointing out that two glucose make a maltose, glucose and a fructose make a sucrose, but what you see here is the dehydration synthesis that kicks off water and makes that glycosidic linkage. Okay? Little quick question for you to ponder: are these two isomers? Okay. This sucralose 600 times sweeter than sucrose, just by the addition of an alkyl by a little alkyl substitution. Okay. Polysaccharides are long polymer chains of sugars, and really long chains of glucose. Here is starch. That's what it's called when you put that long polymer structure together in plants. It can be saved for energy, in which case then you could do a hydrolysis and break off and get one of those glucose monomers. When it's used structurally, as you can see just by a different orientation, then it becomes cellulose, which is something that you have difficulty digesting. Same thing in you. When you put long glucose polymers together, it's called glycogen. Okay? So, monosaccharides are the single unit. That is your monomer. Polysaccharides can be used for starch storage. Again, it's called starch in plants, glycogen in U. If it's used structurally, cellulose in plants, chitin in animals and fungi. I'm a fungi. But chitin is that hard shell. Say when you step on a bug and hear it crunch. There you go. Lipids or fatty acids. Of all the macromolecules, they have a wide variety of different things that they do. Lipids are phospholipid membrane for the protective membrane around cells. Fatty acids are for energy storage. They're the basis of steroids. Cholesterol is the basis of steroids and stuff in your body. So there's technically, unlike glucose or the carbohydrate sucrose or proteins with amino acids, there's not one true monomer. Instead, they're all grouped together because they're hydrophobic and they have many carbon-hydrogen bonds. As we'll look at, one of the things they do is just have a long fatty acid tail, which is just carbon-hydrogen bonds. Okay, so here's the basics. Here's the glycerol backbone. Here's a long fatty acid chain. So this would be a triglyceride because it has 3 long fatty acid chains on it. You've got a carboxylic acid on the end of these. Here's your dehydration synthesis. This is your fat storage. Okay. This one is a saturated fat because it is saturated with hydrogens. It's all full. No double, triple bonds. Okay. And you can see just even structurally by orientation here, stearic acid in margarine. Here is a double bond. Here's an unsaturated causing bending, unsaturated fat in your oils. Okay. Phospholipids, now you see you have the glycerol backbone and you have 2 long-chain fatty acids, but instead of a third, now you have the phosphate group. And that gives you an amphipathic molecule, the hydrophilic head, the hydrophobic tails. You throw those in water, they will spontaneously form a membrane where the tails line up away from the water and the heads line up out. When we talk about cells, we'll talk about the function of the semi-permeable controlling that phospholipid membrane does in being the membrane that surrounds your cells. Okay? But here you can see the lineup. Here's your plasma membrane or example of a plasma membrane. Spontaneously, these phospholipids will do that in water. That allows them to be semi-permeable and controls what goes in and out of your cell. Okay? And here steroids. All steroids are based on cholesterol. And that is another hydrophobic lipid. Proteins, they are pro because they are paid. The monomer is an amino acid. There's 20 naturally occurring amino acids. They create a peptide bond. What you see is you have an amine group and a carboxylic acid group. You have a hydrogen off the carbon. It's sharing. Our carbons love to share 4. And then the R is whatever the side chain is. So the amine group with 1 will bond to a carboxylic acid group, and again in a dehydration synthesis to form a peptide bond. And really what helps control what the protein is this side chain that differs for all 20 amino acids. Here you go. You can see many that are nonpolar. It's highlighted with the what the R side chain is. You can notice these are all nonpolar. Here you have some that are polar. You see a hydroxyl group there. In a lot of there's a sulfhydryl group. There's an amine group. You have some acidic with an extra, you can see the carboxylic acid there. Here's an amine group for basic. Okay. And again, name the type of reaction, condensation, dehydration synthesis, whatever you want to see. But you can see the peptide bond being formed here. And with the 20 amino acids and the variety of side chains and almost the infinite number of possibilities coming when we talk about how proteins are made off of the triplet codon. Proteins are the main structural component of the body. Again, They all have other functions, but it's 30% of the mass of your body, and it really is a structural component based right off of your genetic code. And essentially you have infinite possibilities. So as things adapt and change their environment, and you think about with all living organisms how they adapt, change, how maybe new structures are formed and stuff like that, that can be very simply done just by how the genetic code is expressed and how then the protein is made. And so you can develop and adapt over time structures that allow you to survive in an environment. Okay? Proteins, like everything else, have their function affected by their structure. The way that protein is folded is called its conformation, and there's 4 levels. Okay? The level 1 is just the basic amino acid chain. Okay? That's the primary structure. Okay? Then you can either form what's called a pleated sheet or an alpha helix, and that is the hydrogen bonding of the amino acids together. Just the amino acid, just the primary code, and it's just the hydrogen bonding, as you can see, illustrated here. And then you get to the tertiary structure, and that then results from interactions of the side chain of the amino acid, that R group. Okay. Whether it's covalent bonding, ionic bonding, van der Waals, or hydrogen bonding, or any of the interactions that we have talked about. It is very possible then in that final folding up of the protein that the final product needs more than one protein, like hemoglobin. And that would be your quaternary structure. That is actually, and here is a picture of hemoglobin here, which is actually different proteins coming together to form the hemoglobin structure. Okay. Structure follows function, function follows structure. In anatomy, we call that the principle of complementarity. If something goes wrong in any step along that way, then you won't get the right function. One change in the amino acid of the protein structure can causes sickle cell anemia in red blood cells, and causes a sickle shape, so the red blood cell is not the normal shape. Okay? And here's that example. One change changed how the whole protein came together. Here's a normal red blood cell, here's a sickle cell. This obviously will not be able to bind oxygen and carry it to the cells as it should, and if people are homozygous for sickle cell, their lifespan is generally quite shortened as opposed to a normal lifespan. If they are heterozygous for it, then they actually have developed some resistance to malaria and can live a pretty standard life. But we'll talk about that when we talk about genetic codes. Okay? Proteins may lose their conformation due to pH changes, heat changes. You get a fever, and you can have denaturization. That those bonds hold together in a very short window of temperature. Proteins are also are what enzymes are, and they only work in a short range of temperature and pH. And so, if you're out of that range, then those enzymes are denatured and they can't catalyze reactions. Okay. Still a lot to learn. We'll talk about it in class, but there's these interesting little things called chaperones. You wonder, how do these things fold and get fully corrected with all the stuff in the cytoplasm and the influences and things that could be damaging to the protein. And it appears that these chaperones are one piece of the puzzle in protecting them while they get correctly folded. And we'll talk about those more in class. Okay? They don't do the folding, they just provide a good environment. Finally, let's talk about nucleic acids, DNA. Really, this nucleotide is your monomer. You have a phosphate group, an adenine group, and then one of the nitrogenous bases. And this is your genetic code, which is who you are and what you are, and the order of these things then determines the code for making proteins and stuff. You have pyrimidines and purines. Uracil, of course, is in RNA, not DNA. That's one of the things that separates them, besides the fact that deoxy, you can see the other differential here is there is a missing hydroxyl. Okay. RNA is used to copy the code for DNA, which we'll talk about in transcription and translation. But this is the main functional unit of your genetic code, and we'll talk much more about those as we get into our genetic component of this course. Anyhow, that is a quick review of your macromolecules. Scholar on.